My name is Kaylee Totland, and this is my genetics project on colorblindness. The two categories of colorblindness are red-green and blue-yellow. There are 5 types of red-green colorblindness. Red-green colorblindness has a recessive X-linked inheritance, meaning it is inherited on the X chromosome and is more common in males. This is Glenn. Glenn has protanopia. Normal vision has blue, red, and green cones. His blue and green cones are functional, but his red cones are missing. In an alternate universe, Glenn could have protanomaly. He would have normal blue cones, but some of his green cones are normal and some are green-like but hard to tell. This is often described as malfunctioning red cones rather than totally missing red cones. This is Mike. Mike has deuteranopia. His blue and red cones are functional, but his green cones are missing. Similarly, in an alternate universe, Mike could have deuteranomaly. He has normal blue cones, but some of his red cones are normal, and some of his cones are red-like, but hard to tell. This is often described as malfunctioning green cones rather than totally missing green cones. This is Leo. Leo has blue cone monochromacy. His blue cones are functional, but his red and green cones are missing. This is not to be confused with blue-yellow colorblindness because it follows the recessive X-linked inheritance of red-green colorblindness, and the genetic mutation affects the genes for red cones and green cones, not blue. There are 2 types of blue-yellow colorblindness. Blue-yellow colorblindness is autosomal dominant. Blue-yellow colorblindness can be from birth, but it can also be caused by environment, age, injury, cataracts, glaucoma, and other age-related eye conditions. This is Joe. Joe has tritanopia. His red and green cones are functional, but his blue cones are missing. Similarly, in an alternate universe, Joe could have tritanomaly. He would have normal red cones and green cones, but some of his blue cones are present, but they have limited function. This is often described as malfunctioning blue cones rather than totally missing blue cones. This pedigree is based on an interview of my friend Mary's father, Mike, who has deuteranopia, which is missing green cones. It is an X-linked recessive inheritance. In this interview, I learned that Mike, his brother, his nephew, and his maternal grandfather have colorblindness as well. With that information, I was able to form this Punnett square of Mike's parents, showing that he and his brother both had a 50% chance of being colorblind, which I know from the interview that they are. If Mike had sisters, 50% of his sisters would be carriers, and 50 would not have any of the genes for colorblindness. Blue-yellow colorblindness is autosomal dominant. Joe, who has tritanopia, as shown by the diagram with the J square. Let's say his mother has colorblindness and his father does not. How would that look? Well, we can see that his mother has a 1 dominant gene and 1 recessive gene, and his father has 2 recessive genes because he does not have colorblindness. So Joe had a 50% chance of being colorblind and a 50% chance of not being colorblind. This diagram is imaginary and not true to real life. If blue color monochromacy was not sex-linked but a codominant trait, then a parent who has true breeding deuteranopia, which is missing green, and another parent who has true breeding protanopia, missing red, 100% of their offspring would miss red and green cones, thus they would have blue color monochromacy. Well, how common is it? 8% of males and 1% of females have red-green color blindness, and 1 out of every 10,000 people have blue-yellow color blindness, and it's equal for both sexes. Color blindness can be diagnosed through family history, regular optometrist visits, the Ishihara plate test, which is for red-green color blindness only, or it may be acquired after a diagnosis of an eye disease or other condition. There are also other tests called the Cambridge color test, the Farnsworth-Munsell 100 hue test, and the Farnsworth lantern test, which is used for military and aviation fields. Color blindness is a lifetime disease once acquired, at birth or later. It may worsen over time if it is due to age, but is most often stationary for those born with it. There are no therapies to cure or treat color blindness, but there are enchroma glasses, which can be used for red-green color blindness. The results are limited for those with protanopia or deuteranopia, where one of the 3 main cones are absent. There is a study done on adult male squirrel monkeys. All male squirrel monkeys are red-green colorblind. The study began by proving the monkeys were unable to distinguish red and green. The primates have 3 types of cones that all contain a pigment called opsin. The NIH's National Eye Institute injected human L-opsin into the monkeys' eyes for a couple of weeks, and they were trained to touch a colored patch that was among gray dots. results. Weeks after the first treatment, the monkeys passed the test for all colors, including colors affected by red-green color blindness, and this further proved that the adult nervous system of a primate could respond to new sensory input. Thank you for watching. I would also like to thank Mary's dad, Mike Rosovac, for the interview and fantastic stories, and his family members' names that were used for each example. Goodbye.